Our third grant recipient is Dr. Natasha Chang, Assistant Professor in the Department of Biochemistry at McGill University in Montreal, for her project entitled Inducing Stress Granule Formation in Muscle Stem Cells to Treat DMD. Dr. Chang, I'll pass it over to you.
Thank you very much, Lisa. Can everybody hear me?
Yep, loud and clear.
Okay, good. I'm just going to share my screen. Okay, so hello everyone and good evening, and thank you for that introduction. It's really my great pleasure to present our Jesse's Journey research project to you this evening. And I want to start by saying that it really is an honor to have our work recognized by the Research Funding Advisory Committee and the Duchenne Family Advisory Committee. So I first want to say thank you to the committees and the reviewers for really taking a chance on a young lab. And we are really excited to have our work funded so that we can make some interesting scientific discoveries and contribute to the Jesse's Journey community by finding new ways to treat Duchenne. And of course, thank you to the entire Jesse's Journey community, the donors and the volunteers for making it possible to fund these exciting research projects. So in my lab at McGill University, the way that we approach Duchenne muscular dystrophy is we are interested in understanding how the lack of dystrophin, which you've heard is the genetic cause for DMD, affects a group of specialized adult stem cells known as muscle stem cells. And you've heard about these from Dr. Bengston's talk. And so we also call these cells satellite cells. And they're pictured here on the top right-hand corner. And this is really the first description of the muscle stem cell in 1961, when Alexander Moreau observed a satellite cell that was sitting on top of a muscle fiber. And that's the cell that's pictured here in this cartoon. And from this picture, you can appreciate that the muscle stem cell is a rare cell that resides within the muscle. And these cells have the responsibility to make new muscle cells in response to injury to repair the damaged myofiber. And these are the cells that are responsible for sustaining the regenerative capacity of our muscles throughout life. And so in my lab, we use different experimental models to study these muscle stem cells and the regenerative capacity. And so these are just some examples of the techniques that we use. On the top here is a picture of a single myofiber, and in the middle of this myofiber is a muscle stem cell that has just divided and undergone a cell division to generate 2 stem cells. We can also culture these muscle stem cells in the lab, and we can induce them to differentiate, which is when they convert from a stem cell to a mature muscle cell. And we can visualize the fusion of these cells into myofibers, as shown in this bottom left panel. And finally, we can also make use of mouse models to study muscle regeneration, and we can use genetic approaches to alter muscle stem cell function, or we can use muscle stem cell transplantation methods to look at the impact of the transplanted cells on muscle regeneration within the mouse. And on that note, we also use mouse models of Duchenne muscular dystrophy to assess how stem cells are affected in this disease. And so I became really interested in muscle stem cells and their role in DMD during my postdoctoral fellowship in Michael Wernicke's lab at the Ottawa Hospital Research Institute. And Michael's lab really moved the field forward in 2015 when they showed that dystrophin is also expressed in muscle stem cells. And so during my postdoc, I found that dystrophin in muscle stem cells controls a cell signaling pathway in satellite cells that is important for muscle stem cell differentiation. And so this process of a stem cell generating a muscle progenitor, or a cell that will differentiate into muscle, is being regulated by the dystrophin protein complex that's shown here. And what we found was that this has really important implications in muscle stem cells in Duchenne muscular dystrophy. And so what is becoming more evident as the research field moves forward is that muscle stem cells are also impacted by the loss of dystrophin expression within the stem cell. And so I already mentioned the work from Michael Wernicke's lab demonstrating impaired cell division in dystrophic muscle stem cells. And we further showed that this results in impaired gene expression of muscle differentiation genes. but work from other labs have also shown that muscle differentiation is impacted in dystrophin deficient muscle progenitor cells. And newer studies show that the mitochondria in muscle stem cells are also not functioning properly, and that the muscle stem cells have altered metabolism, and they also go into this altered cell state known as senescence, where the cells can no longer enter the cell cycle to divide. And so the way that my lab envisions Duchenne's muscular dystrophy is really that it is a two-pronged disease, where the absence of dystrophin in the muscle tissue leads to muscle fiber instability, resulting in myofiber damage, but also lack of dystrophin in the muscle stem cell leads to stem cell dysfunction. And these defective stem cells also contribute these disease-causing entities to the muscle fiber and contributes to DMD pathology. So we really see it as both a muscle disease as well as a stem cell disease. And so the rationale for our project came from discussions with Imed Galuzzi, who is my colleague in the biochemistry department at McGill. And his lab has done very extensive work looking at ways to improve cachexia, which is a condition of severe muscle wasting that is frequently observed in cancer patients. And so using both mouse and cell models, they found that a certain class of drugs can improve muscle differentiation and prevent muscle wasting. And the way that these compounds act is through the formation of these cellular entities called stress granules. And so we asked a very simple question, which has turned into a really interesting research project, which is, can we use the same strategy in dystrophic muscle stem cells to improve their regenerative potential? And if we correct these stem cell defects in DMD muscle stem cells, can this lead to a potential treatment for DMD? And so these are some of the very first experiments that we performed in muscle progenitor cell cultures. We took cells from both normal as well as DMD mice, and then we performed differentiation assays using varying dosages of these drugs. And what we're really excited to see is that we can improve muscle differentiation at very specific concentrations in both healthy as well as dystrophic muscle progenitors. I want to acknowledge and introduce you to the research team that is behind this project. I already mentioned Imed Galuzzi, who is my colleague and mentor, and he's also a co-applicant in this project. And this work is really spearheaded by Romina Filippelli, who is a master's student in my lab, and she'll be staying on in my lab to continue to do a PhD. And she has been helped by Shirley Lee, who's an undergraduate trainee in the lab. I also wanted to point out that this funding will provide financial support so that we can recruit an additional PhD student, a postdoctoral fellow, as well as summer students in my lab to move this project forward. So thank you also for contributing to this very important aspect of training the next generation of young scientists. I also wanted to acknowledge my partners on this project. The Stem Cell Network has been instrumental in helping me set up my lab and has really provided excellent resources to navigating the translational research landscape within Canada. And the McGill Regenerative Medicine Network has also provided a lot of support and provided a very collaborative network for regenerative medicine research at McGill. Lastly, I want to thank the members of my lab, and you can follow us on our website to see what we're up to. And thank you to my wonderful collaborators that support different efforts within my lab. And of course, research would not be possible without funding. So I'm really very thankful to the Jesse's Journey community, as well as other agencies that fund our research. And I really thank you for this opportunity to share our work with you.